From the Donald Danforth Plant Science Center, HEC-TV, Boeing, and the Danforth Center are proud to present Conversation, a discussion about the intersection of plant science and baseball.
I'd like to welcome everyone this evening, friends and guests of the Donald Danforth Plant Science Center. I'm Molly CLINE, and I'm chair of the Conversations Committee. I'm a plant scientist by my academic training. I'm retired from Monsanto. And I feel very privileged and honored to be part of this conversation this evening. The Danforth Center, its mission, improving the human condition through plant science. Improving our food crops, our fuel crops, and doing that sustainably. And enhancing the St. Louis region, putting us on the map globally for plant science research. That's really something. Tonight's conversation, I know we all want to get into it, we can't wait. Let's talk about the intersection of plant science and baseball. Okay. Well, I want to thank the Friends Committee and the Conversations Committee for hosting conversations. And guess what? This is the 10th year, and we're still not finished talking. There's going to be a lot more conversations. I'd also like to acknowledge the Boeing Company for their 7 years of sponsorships of these conversations. Tonight's session will be recorded for HEC or by HEC-TV and aired starting on November 24th, a Sunday at 5 p.m. And please take note that previous conversations you can access through the Danforth Center website. Our panel tonight, I'd like to introduce VIJAY CHAUHAN, who is an entrepreneur in residence at the Biogenerator here in St. Louis. And our distinguished panelists, Dr. Ivan Baxter. And he is a USDA ARS research scientist. He is also an assistant member and principal investigator here at the Danforth Center. Would also like to introduce Chris Correa, who is with the St. Louis Cardinals, and he's director baseball development. Well, I'll tell you, as a plant scientist, I am all ears tonight to hear about how numbers, statistics, data, how do they help predict a winning plant and winning baseball players? So, okay. VIJAY, it's all yours.
Molly, thank you for that wonderful introduction. I am so excited to be back here. I'm still a moderator in training. This is my second gig. So I need your help. And with a topic like this tonight, please feel free to put out the questions that you've always been wanting to find out from Chris and his wonderful organization, as well as from Ivan. I got to tell you, when I was preparing for this, I had a chance to talk to both of them. And with Ivan, I was obviously going to focus on plant sciences and genotyping and phenotyping and how that translates into success and what the Danforth Center does. And I found myself having to always go back to baseball because that's where Ivan wanted to go. And actually did a very elegant job of connecting the two. So if you hear baseball from Ivan, don't be surprised. So I'm really excited to have both of them here. We are here to talk about and have a great fun conversation, but also an important one. So in the year 2000, there were 5 billion people on this planet. As of this year, we have about over 7 billion people on this planet. And the projection is that by 2050, there are going to be over 9 billion people. To be able to feed this population of 9 billion people, we are going to need to double the output of agriculture. And what's even more challenging is that we're going to need to do that with lesser land and lesser water. Why? Because these 2 billion people are going to need a place to stay, which is land. They're going to need to send their kids to school. They're going to need parks. They're going to need shopping malls. They're going to need baseball stadiums. So they're going to need land and they're going to need water. And here is agriculture going to have to give up all this land and yet double the production to deliver that. That's a huge, huge challenge. And the Danforth Center, its leaders, its scientists, all you supporters are dedicated to the mission of making this huge challenge happen. They're not doing it by themselves. They're doing it with a global community of public and private institutions. But that's what this center is dedicated to doing. And you all, by being here tonight, are signaling your own support for that mission, and we all thank you for that. So that's the big and important mission. And so we will be putting that as the backdrop for the conversation we're going to be having tonight. So we're going to be talking about baseball in plant sciences, and you're going to be wondering, one is about entertainment, and the other is about research. What could they have in common? Well, you'll find out from the experts today that in both of these fields, digital technology is transforming how these institutions work and run. The amount of data that is being generated, that is being captured, that is being analyzed is transforming how decision-making and how these institutions are able to do the work that they do. So with that, I'd like to say welcome to Chris Correa from the Cardinals. He's the Director of Baseball Development. And Ivan Baxter, the USDA research scientist and the principal investigator at the Danforth Center. So welcome, gentlemen. And please don't be bashful with the questions. I know they're going to be a way for you all to write your questions and hand it to somebody. And I'll do my best to bring up the questions that fit with the talk tonight. So Chris, why don't you tell the audience a little bit about what does a director of baseball development do at the Cardinals?
Sure. Well, first I want to thank the Danforth Center and everyone here for inviting me to talk. This is a really important place, and I'm happy to contribute to this conversation. So my role is I manage a small research and development group. We are 6 people, which is small by a lot of industry standards, but by baseball standards is quite large. And so our responsibility is to do the analysis and leverage technologies to support data-driven decision-making in all areas of baseball operations. So, for example, which player should we trade for? Which player should we promote from a minor league team? Which player should we draft or sign? How should we prepare for an upcoming opponent? These are the kinds of decisions that get made all the time, and our department's responsibility for bringing in data and assisting in those decisions.
So you heard him say research and development. Would any of you have thought that a baseball team has a research and development department? And yet, it is fundamental to the success of the Cardinals team, and you'll hear more of that along the way. So Chris, tell us about your background. So how did you, what were you doing before, and how did you come to the Cardinals?
Sure. So before I joined the Cardinals, I was in the PhD program at the University of Michigan in the Department of Psychology. And you're all probably wondering how I got this position from there. Well, when you're a graduate student doing studying research methods and statistics, you'll often sort of encounter the same old data sets to sort of learn different techniques. And so, as an example, Ronald Fisher in 1936 popularized a data set of iris species. And so you take the petal length and width and come up with some multivariate approach to determine which species was which. And you'd sort of work on these kinds of data sets over and over again. I didn't realize at the time that it was like a crude form of phenotyping, but apparently it was. So we'd work on these kinds of data sets, and I would always wonder, well, okay, this is kind of interesting, but how can I make this more fun or interesting for me? And I would try to apply what I was learning to baseball sort of data sets that were available. So I was a baseball fan. My playing career ended in high school. So I started applying these different research methods and statistical techniques I was learning to baseball. And the Cardinals had an opportunity that they advertised So I worked for them part-time in 2008, and in 2000, while I was trying to finish up my PhD. In 2009, they extended me a full-time offer, and I didn't quite finish the dissertation, but I decided that it'd be cool to work for a baseball team. So I tried out for a year or two, and I'm still here.
So thank you so much for that background. Ivan, tell us a little bit about what you do here at the Danforth Center.
I drink coffee here at the Danforth. No, my lab studies how plants take up elements from the environment. So your iron, zinc, magnesium, calcium, everything that's, a lot of things on your vitamin bottle. How do the plants take those up from the environment? And we use that to figure out how plants are adapting to their soil environment. And so we have a high throughput profiling system that allows us to measure 1,700 samples a week. And in addition to that, I'm on part of the team and I'm on the committee that has developed the phenotyping facility here, the Bellwether Foundation phenotyping facility. And that's where we're using even more throughput and more ways of analyzing plants to try and understand similar questions and more.
So we'll come back and talk a little bit more about that wonderful phenotyping facility that you have here and how it relates to baseball here in a second. By the way, it does relate, you'll find out here. So Chris, why don't you tell us what area of the Cardinals' activities has been most transformed by the data analytics approach that you have been integral to developing?
That's a good question. A lot has changed over the past 5 years or so in terms of how we make decisions. I think probably the most striking contrast is in the annual draft every June. So every June, baseball teams have the opportunity to draft 40 players from the amateur ranks and bring them into their organization. And the way this used to work was scouts would go out and see players, they'd evaluate them based on what they call their tools, and they'd grade them out, and they'd get into a room in June and just sort of debate and argue about which player should be ranked where. So every team would have a master board, and they'd sort of pick from that board which player they were going to select in the first round, the second round, and so forth. That's how it used to be, even as recently as 7 or 8 years ago with the Cardinals, and it's still that way for a lot of organizations. And now we just have a much more data-driven process in place where we consider the performance history. We sort of quantify that and assign it a value to predict their future performance. We look at medical reports. We quantify that and assign a value to help us make a decision. We look at the scouting reports. They're still critical to our decision, but we sort of normalize them and try to find what the right balance is. Should we give the scouting reports 60, 70% weight, 40% weight? And so it's all very much, it's all a lot of digitized data, and we're trying to find the optimal way to balance all that information. And we assign a value to every player, and that's how we rank players now. So it's a lot different than a bunch of baseball veterans coming into what they call a war room in June and decide how to rank players that way, just through sort of the same kind of debates some of you might have about which player you like more or who deserves the MVP tonight.
So we're all dying to know, Chris. We've all seen the movie Moneyball, right? There's got to be a Moneyball moment in the Cardinals organization, right? So Chris, can you share with us an example of how, using a traditional approach, there was this bias towards this player, and then when you apply the numbers, how that changed?
Sure, I have to be careful here. I think there are a lot of examples. If you look at the players we've drafted, most players, when they're in college, if they're any good, they get drafted their junior year. That's the earliest they're allowed to be drafted, and they'll finish up school, some of them in the off season. And I think what you might see in our drafts, if you look at them, is that we've had a lot of successes with players who were drafted their senior year, so after they've graduated from college. And the reason is because there was always something about the player that caused scouts to bypass them. So they weren't even drafted; they were sort of overlooked their junior year, and based on their performance and some of the analytics that we do, we were able to identify them as players we should bring into the organization, even though they were sort of seniors and sort of these second-rate players. And so, I'm proud of some of the work that we've done, and the scouts deserve a lot of credit because they'll go in. Sometimes we'll identify a performer, sometimes they'll already know about him, but they'll go in and take those players seriously. And sometimes it's been as simple as Alan Craig was playing shortstop in college, and he's just not a shortstop. And so I think from a scouting perspective, you can sort of focus on what players can't do well. And he could not field very well as a shortstop. But he could do a lot of other things well. And so, once we're able to sort of balance all that information, we can identify him as a player that's worth drafting. And we took him as a senior out of Cal, and he's been a pretty productive player. There are others like that that have not been as successful, but that's sort of an example of the kind of those sort of moments where sometimes we disagree, and there are other times, I promise you, when the scouts are right and I'm wrong. So,
well, he handled it with great diplomacy, didn't he? Nice job. I didn't even prepare him for this question. So, Ivan, you can hear from Chris, it's, these guys are all focused on trying to pick winners. Y'all are trying to do the same thing in the plant sciences area. Can you share with the audience here how you go about doing this in the new world of genotyping, phenotyping, with the great technologies that you have here? This idea of picking winners, what does that look like?
Where we are, there's a bunch of different technologies that we can bring at that question. Some of it is these great advances in sequencing the genome, which is the DNA that is encoding the plant, and some of it is our better ability to measure how the plant is reacting to its environment. And so we have lots of new technologies to sequence and detect the genome, and we have several new facilities that allow us to really look at the plants in new ways. And those are things like the Bellwether Phenotyping Facility's imaging, and there's several different kinds of imaging. And there are microscopes that can allow us to see more in depth what's going on at a single plant level. And then there are ways of characterizing things at the biochemical level, like what I do and our metabolomics core does, where you can ask what's going on molecularly for the plant. And then we have to bring all that together to decide whether or not to draft Michael Walker. I mean, to pick the right plants that we would go forward. And can we take some basic knowledge and then move that out?
Got it. So what you're hearing here is there's such a similarity of the mission, which is to find winners. The process is about using data and analytics. The tools are unbelievably sophisticated technology. And one of the things that you may want to share with the audience is how special is the phenotyping facility? How many places in the world have this kind of facility in a public research setting?
There's only one that has what we have out there, and that's here. Mm-hmm. And so that's pretty cool. There are a couple others that are doing things in greenhouses that have some similarity, but we're the only ones who can control the environment. So we have a controlled environment chamber that can fit 1,100 plants. And so we can do really well-designed statistics that R.A. Fisher would be very proud of when we set up our experiments. And because we can control the environmental variables, we have a lot more ability to set up those experiments correctly to figure out what the effect is of those variables. And so that's what makes us unique. And then we can look at all those 1,100 plants over a couple days, and we're going to be able to see not only what they look like, but we can also see how well they're doing photosynthesis with one system. And we have another system that can tell how much water there's in. So it's sort of like, not only can we take pictures of you, but we can take a quick x-ray of you and get a feel for how you're doing, and we can do that very rapidly.
Right. So the picture I took away from this is like putting a plant and putting 100 probes on it and being able to measure what's happening with it 24/7. That's an incredibly difficult task for any human being to do. Here's a system that does it on 1,100 plants, you said. Mm-hmm. And so that's an enormous amount of information, and then the scientists can look at that, go back in time and say, okay, the plant really took off and grew like heck in this time period. What was going on at that time period? and then be able to understand what caused it. Is that a
That's definitely part of it. The time component is something that's really new for us to start integrating into our analysis. Yeah.
Excellent, excellent. So I'm going to bounce back to you, Chris. How
Sure.
did the Cardinals get into this philosophy of using analytics and statistics and a whole new way of doing what you do?
Sure. Well, one of our owners read Moneyball, and that helped. But in all seriousness, I mean, I think maybe as early as 10 years ago, our ownership group recognized that the economics of the game were changing, and they needed to devote more resources to the draft and player development, and that was going to be a real focus for us. And one of the tools, or one of the approaches they used to try to improve the draft and player development was to bring in the use of analytics. And so that's sort of where the seed was planted, and sort of driving sort of draft room decisions, which I've already talked about. And since then, it's sort of propagated to influence a lot of decisions regarding player development, or even the major league transactions. And so there are a lot of, really, it was a lot of ownership from up top in terms of our ownership group, general manager John Moselak. I mean, they've really bought in and have been really supportive, and that's sort of how we've gotten to the point where we've gotten.
So big culture change.
Yeah, very big culture change. Yeah.
There's a traditional way of doing things, and here's a new approach. Looks good on TV and in a book, but
Yeah.
how do you pull it off?
Yeah, I mean, there are a lot of veteran baseball people, former players or coaches who are still in the organization, are really valuable members of the organization. And I think it's been an adjustment, but I think it's been a learning experience for both them and for me. I think when we're at our best is when we're learning from each other, and I think we've definitely developed that culture now. It's been sort of slow to start, but and every year it's getting a little better. But that's one of the parts about my job that I really like is, explaining to a former big league pitcher like Alan Bennett sort of what the data says about a particular player, or sitting behind home plate with a former manager like Mike Jorgensen and understanding how he sees the game and how maybe a win probability model might view the game differently. And so that's it's been a big culture change, but it's really one of the parts I find most rewarding about the job.
For some of the old timers, you're throwing language and
Yeah.
vocabulary that's pretty different, isn't it?
Yeah, it's different from what they're used to, but to their credit, they're willing to learn, and they ask a lot of good questions that help make our models better, frankly.
So Ivan, what impact has the phenotyping facility had at the Danforth Center in terms of giving the scientists a view of the new tools that are here and the kind of impact that you can have and how it furthers the mission of the Danforth Center?
Well, as we've just got it up and running, the immediate impact is one of great fear. How do we take this tool and make it useful? But it's I think from the second that you find out that you could do something like this, for a lot of us, it opens a new world, because there's a lot of questions that you didn't think you could answer before, and so I think. Just to give you like a more concrete example instead of rambling, we don't know very well what the effect is if you have a little bit less water for a plant at each stage of its life cycle. As it's making a decision, do I put out another leaf here or another leaf here, or do I go a little higher and then put out a leaf there? And it would be very hard to do that kind of experiment conventionally, where you send out all your people in your lab to go measure the length of the stem and when the leaf came out, and they like to go home on weekends. It's a hard thing to convince them that we should be in there, and nobody really wants to give you the money to have 20 people sitting in the greenhouse every night asking those questions. But it's an important question. There's 2 years ago, we had a tremendous drought in the Midwest, and if we're going to improve the crops to use water better, we need to understand that process and how the plants are dealing with that stress. And so now we have a machine that can control the water very carefully for 1,100 plants. And you can say, okay, you give these ones 20% less water and see how they respond to each other. And so that's the kind of questions. And now we just got to figure out how to extract all that data. And I think one of the things that, obviously baseball's awesome and it's cool, but one of the cool things about what Chris is doing is that they're actually figuring out ways to use the data. It's one thing to collect it, it's another thing to use it. And we have a lot of good ideas of how to use it if we can just understand it. And it's that huge meeting of those things that we're really starting to work on now.
That's a great example of the kind of things that this new phenotyping facility is doing here. How has that impacted the morale, and how are people thinking about this?
Well, there's a lot of excitement going on. There's a lot of, the postdocs are really energized. They're all thinking of new ways to do experiments, and there's just it's a lot of anticipation to really get on there and do your experiment. So it's really easy to get. It's a lot easier to get people to come work here, I think, when you can sort of show that, or if you show the video that you guys saw out there, if you're in a talk, all of a sudden people are asking you questions about it and they're curious. How's it working? Yeah.
So you're trying to predict based on data. You're trying to make predictions based on data. Give the audience a sense of the volume of data, Ivan, that y'all are working with.
Right. So every time a plant goes through the system, the imaging loop, we can collect 10 to 20 images on that plant. And that's just the beginning, because that's the base data. And then you start turning that into models, and you can collect more data off of that. And so those are big images like you'd get off a really good camera. And so you're collecting thousands and thousands of pictures every week that you have to analyze and track and process. And that's just one system at the Danforth Center. There's also other systems that are creating data just as large all the time.
So if you want to measure it in terabytes, megabytes, gigabytes,
it's terabytes. We have lots of server space here, and I keep my music there too.
So in preparing for this talk, I was reading about data and big data, and what I realized is apparently every day we as a human society are generating 2.5 quintillion bytes of data. So that is, a quintillion is 10 to the power of 18. So what does that mean? It means that if every person in the world today was given a desktop or a laptop computer with nothing on it, by Saturday morning, all 500 gigabytes of it would be full for every person on the planet. That's the amount of information that we are filling. They would have to come back on Saturday morning and say, I'm full, give me the next computer, because I got to take all this data that's being generated. So we are generating 2.5 quintillion bytes of data every day. And so when you talk to folks in the Plant Sciences Center, you begin to realize how much is biology and plant sciences, how much is information technology and database management and software programming and so on. So we'll be coming to that here in a second. But there's a more interesting inquiry that people have. They want to know, Chris, did your work have any influence in picking Michael Walker?
Well, frankly, we were surprised that Michael Walker fell to us in the draft. Our scouts viewed him very highly, and he was the top performer available to us in the draft. So, he was from Texas A&M, and he.
Are you going to be drafting Johnny Football then?
Probably not. So yeah, I guess it played a small role, but I mean, he was well liked by scouts and everybody in our organization, and we're happy to have him.
So, Ivan, tell us about the kind of people that are on your team that are working in these kinds of projects. A little bit about the makeup, their background, the experience.
So we've got a lot of people working on it. We have a facilities manager, and she's the problem solver. So she's got to make sure that the carts run on time and that the jobs get scheduled and that everything's working. And then there's a computational core that helps out with handling all the data and moving it all around. And then there are postdoctoral researchers whose job is to really try and do the science with all of this. And they're just beginning to start to look at all the ways that we can analyze the data. And so as part of that, we've been talking as a group to computer scientists at Washington University. We've been talking to biomedical engineers at Washington University to start help us build these images into models and models into data. And then from there, we can take the data, compare it with the genetics, and then the idea is that we can identify parts of a plant's genome that are associated with the models and the data, and that's going to tell us how the plants are growing and how we can use that knowledge to improve a crop.
So you've got at least 4 or 5 disciplines of people involved in a project like this?
Probably even more because our postdocs have multiple disciplines that they're bringing in together.
Yeah, so this is a highly cross-functional, multidisciplinary team.
Yes.
How do you guys sort out who's in charge and which discipline rules and so on? Is there a...
Oh, the PI's rule. That's the way we always do things at the Danforth Center. It's a lot of it's a lot of talking and coming together and figuring out. It's more problem driven than it is discipline driven. So we talk about what we're trying to do, and here's the hurdle. We don't know the best way to construct a 3-dimensional model with 15 images. And there are 5 or 6 different ways to do it, and we're not sure which is the best. And so then you have to, if that's not someone's expertise, you have to go figure that out. And I think that's the way we let the experiments. And we have questions that we want to answer with the system, and then you let those drive your decision making.
So teamwork is a critical...
Teamwork, yeah.
Yeah, ability to work in teams, put
Right.
your ego aside.
Yep, and know who's going to be where in the rundown. Sorry. You have to... Sorry.
So Chris, tell us about the teams that you have in your organization, what their background skills are.
Yeah, so our research development team is it's interdisciplinary, and I like the way that Ivan put it. It's really, all of our discussions are problem-based, so whether your background's in applied math or statistics or in computer science, everyone has different experiences and tools that they can sort of bring to solving these problems. So it's an interdisciplinary team. There is no program graduate program in baseball that feeds us or anything like that. So we just try to bring in people who have interest and passion for baseball, but also some unique skill set or ability to break down problems in a new way that can help us.
So Chris, tell us about how difficult it is to get one of these jobs in the Cardinals organizations.
There aren't a lot of these jobs out there, and I think that'll be less true over time. Probably at this point, most teams have at least one person doing this kind of work. Maybe half a dozen teams have groups between 5 to 10 people like we do doing so. And there's still a lot of teams that don't really put many, if any, resources to this. And I think that'll change over time, and there'll be more of these jobs available. But it's definitely a low supply, high demand issue.
But there are also lots of people who are wanting a job like this, right?
Yeah, there are, and we get lots of applications. We don't really post positions, but we still get applications. And there are a lot of people with passion for baseball and a lot of different interesting skill sets that they want to apply.
I don't mean to flood your inbox with potential candidates from this audience tomorrow.
That's fine. We're used
But
to it.
I remember meeting somebody who was working at the Cardinals organization. He was a scientist, a PhD at NASA, working on the Jet Propulsion Laboratory in California. And he was very keen to work with the Cardinals, and he had 85 phone calls, 45 emails, and 10 attempts to just say, I'm going to fly to St. Louis to meet, before he actually got a job. And this is a person with a tremendous amount of experience and credibility. So it just shows what kind of people are interested in a job like this.
Yeah, diligence matters a lot, persistence.
We don't have that problem.
Yeah.
I think we have a, I mean, if you have the skills, we certainly have a supply problem. I think we're always looking for people who can fit into that data-driven, problem-solving approach to what we're doing, and I think that's a real key skill. But that means you have to be able to look at the data. You have to be able to move the data around, and so we're definitely looking for those types of people. So if people don't want to do baseball phenotyping, they want to come do plant phenotyping.
I'm going to start redirecting.
There you go. Send them our way.
So we've got an HR strategy here
There we go.
of talent sharing. Jim, be careful. You might have a baseball academy here before you know it. So people want to know, what is phenotyping? Can you just break it down into, and genotyping into something that people can relate to?
So phenotyping is really the ability to understand everything that's going on about the plant, mostly image-based. So the idea of phenotyping is that we can understand the full plant. So it's, the ideal one would be if we have the ability to measure everything we can about the plant. That would be the plant's phenotype. But in the context of our facility, it's really an image-based recording of the plant's status. So.
And tell us about genotyping and the relationship between the two.
Right. So your genome is your DNA. It has the instructions for what's going to happen to your as you grow and develop. But and right now we have the genome of humans or of several of our plants, but that's just one plant's genome. You can think of it as a book. It's full of letters. We're just not really sure what are the words, what are the paragraphs, what are the chapters. And every species, every line that we look at, every human has a different book. And we need to be able to read that out. And so what we're now able to do that we weren't able to do 10 years ago is get lots of these books. We can collect a lot of genomes. But we still don't know how you get to the plant out in the field. And a lot of it is how the plant is reacting to its environment. And so a lot of what the plant's genome codes for is how do you respond to the environment as you grow to survive where you happen to be planted.
So it's really the nurture-nature part of the equation here?
There is a big component of both of those into what you see at the end. And I think that's probably a good thing to think about when you think about baseball. When they draft someone, they have to know what's in the player's DNA, but they also need to be able to say, we're going to convert Rosenthal from a shortstop to a pitcher because we know the environment that he needs to have to be able to strike out the whole side on 12 pitches, right?
So, now we get it.
That was pretty cool, by the way.
Now we
I
get
agree.
it.
Yeah. So it really is. There's what we're trying to understand is how the plant takes this string of letters and survives and thrives in this environment, and maybe not in this environment.
Got it. Chris, tell us about some of the technology that you'll have in place at the Cardinals that really helps you do what you do?
Sure. Well, after spending some time here, I want to take one of these phenotyping facilities and put it in the Dominican Republic and just start running players. see what we can learn. So we'll maybe talk about that after. I think probably the biggest change over the past 5 years or so is that we're less focused about the results of a baseball game. So how many hits you achieved, how often you got on base, and so forth. And we're really trying to understand how you got to that, how you got those results. So, for example, we have via imaging technology or via Doppler radar, we have measurements of every pitch ball and batted ball at the major league level, and many times at even the minor league level or even collegiate games. And so once we start understanding how this kind of information can help predict future performance, that will sort of give us a competitive advantage that we can sort of carry forward, in that we're not just looking at sort of your past performance in terms of whether you struck out somebody on 12 pitches, struck out the side on 12 pitches, but we can actually see, okay, this is how he did it. And some ways might be easier to sort of carry forward in the future. Some might be predictive of future improvement or decrease in level of play. But that's sort of where we're at now, is that we're trying to understand all this ball tracking information that's coming in and what to make of it. That's been the biggest change over the past few years.
So one of the things we were dying to know is, what's the one question that Mike Matheny has for you? Chris, why can't you tell me about this? What is it? What's it that he wants to know?
He has a lot of good questions about game strategy that sometimes I don't really have a good answer for. When to bring the infield in, for example. Well, we don't actually know exactly where fielders are standing yet. We have algorithms that, based on the game state, can predict where we think infielders are standing, but we don't actually have a precise measurement yet. And so I can't really tell him, like, if you move a fielder in 10 feet, for example, how does that increase the probability of your pitcher allowing a hit? So we have some way, we can estimate it, but that's an example of something that the technology isn't quite there where we can track every player in the field for every game.
What's the game state?
What does that mean?
Well, how many balls, strikes, runs,
Okay.
there are
Yeah.
going on.
And what's the one question that he's been wanting to have an answer for, that you're getting ready to have an answer?
He has a lot of questions like what I just talked about infield and outfield positioning. And so, are we doing a good job of positioning our outfielders before every play or before every pitch, really, it changes? And so these are the kinds of questions that we've been trying to focus more on and give him a good answer regarding, look, we're tracking down more balls than the average team, or we're not, and understanding sort of the interaction between the player abilities and where they're being positioned pitch by pitch. And that's something he has a lot of questions about. He has a lot of great questions about a lot of baseball topics, but that's one in particular that comes to mind.
So Ivan, now you're in the hot seat. What questions does Jim have for you that you're out there trying to figure out how to answer with all this awesome infrastructure and capabilities you'll have?
When are you going to get the next grant? No, I think what the next big question is really that idea of how do changes in the environment change the decision-making a plant goes through as it's growing. So when to put up a leaf, how long to make that leaf, and and how does that change when the plant loses water when it's got 2 leaves versus when the plant is about to set seed, and those types of things. So the timing and the dynamics of how plants respond to the environment is something that we can maybe even start to ask, which is pretty cool.
And because of that, what will be possible?
Well, then the hope is that you can start to predict which plants of your breeding program will work better in certain environments as soon as you can predict the weather, which I'm sure is just around the corner. You guys will do that.
Yeah, right.
But not just the weather, but if you know that this is what the climate is and the soil is in this location, then you should be able to make plants that are better able to withstand that environment as you go forward.
So how many teams in the baseball, in the MLB, have this kind of R&D capability that the Cardinals have?
It's hard to know because we're all paranoid and very secretive. But based on watching how other teams behave and knowing some other people, I would guess maybe a half dozen teams have similar investments in research and development, and many other teams, most other teams have at least some investment. So I think I mentioned before, that's growing every year. So.
But you are definitely in that very small group of teams that's into this in a big way.
Yeah, and like I said, it's hard to know exactly how many teams or where we rate in terms of our investment, because we all want to believe that we have some competitive advantages that other teams don't. But I think certainly our investment, and probably more importantly, the way that sort of our research is actually integrated into decision-making processes by the senior leadership is probably that's something we've been working on for a long time, and I think it's advanced relative to what other teams are doing right now.
And Ivan, you can be as proud as you want to. How about the Danforth Center and its where it sits in?
Oh, I think we're pretty high up there. I mean, there's very few places that have what we have. Even before phenotyping facility came out, I would bring my friends here, and you walk into the plant growth facility and see all the chambers that you can do experiments with. And they get a little jealous. It's really a unique place. We have some amazing scientists here, and you don't have to be here very long before a lot of people get sold on wanting to come here. It's a top flight place, and we've got some things that no one else has now. Like we can talk about the Cardinals.
So now you can see why these organizations have so much in common. They're the top of their game. They invest in R&D. They have tremendous infrastructure. So one thing that I wanted you all to share with the audience is, what advice do you have for kids these days about the kind of education and skills that are going to be of value in these new economies, in this new age that we're all moving to? It has so much of digital information, big data, analytics, etc. Chris, you want to?
Sure. VIJAY, you've already mentioned how information that is being captured and collected is just increasing at unbelievable rates, really. And so I guess my advice, in addition to sort of doing well in school at the things that are going to matter, like math and computer science, my advice is that anyone, no matter what you're interested in, whether it's plant science or baseball or some other domain, like film or some other science, I feel like there's somewhere there is publicly available data that you should get familiar with and start asking questions and doing some inquiry-based projects where you're actually learning how to manage and use the data to answer questions. I think that's sort of the number one thing I would encourage any teenager really to do, because there are a lot of publicly available data sets in all sorts of domains now, and there are a lot of freely available tools to sort of manage and do something with that data. And I think, like I said, no matter what kind of industry you're looking to get into or what really interests you, there's going to be some way that you can get some data and start playing with it.
Excellent. Ivan, what would you say about that?
Spend more time on Facebook. No, that's not. Actually, I wouldn't say that. I mean, in addition to what Chris said, and I think that's absolutely true for scientists as well, and I think we are moving towards where everything is digital, and you answer questions. You may set up experiments in the lab, but you are going to get the answers on your computer. And so you have to be comfortable with data. You have to be comfortable with thinking about data and analyzing data and new methodologies. I think you do have to be able to work with other people, because nobody is going to have all the skills that we need to actually solve any given problem. It's we're no longer where the physicists were writing stuff on an envelope, and that was how they did research. We have to work in collaborative teams, and you have to work together, and you have to be able to communicate across disciplines, and frequently that's about data and methods, and how do you bring those together. And so I can't emphasize enough the ability to deal with data, but it does matter how you interact with people, because we are all in this together when we start to ask these questions.
Yeah, I agree with that.
So thank you very much. We are out of time. I wanted to really thank Chris and Ivan for breaking it down from very deep science to what people can understand. There's a lot of overlap between baseball and plant sciences, as you can see. And the thing that I'm most excited about is that we have in St. Louis 2 premier institutions, one in plant sciences, one in the game of baseball, and there's so much in common. So thank you very much. Jim?
Thank you, Vijay.
That was fantastic. Thank you so much, VIJAY. VIJAY, this is your second time hosting. I don't know if this is equivalent to the late night television host change that's coming up here, but thank you so much for doing this. Chris, thank you for coming over across town. This is the second time we've had Chris here at the Danforth Center. We had Chris here a couple of months ago for a scientific symposium. We had a very multidisciplinary couple of days worth of scientific presentations from neuroscientists, from epidemiologists, plant biologists, and the baseball guy. And he hung right in there with the absolute best science in this really fast-changing and complex field of phenotyping. And Ivan, thank you so much for sharing your insights. Ivan's a terrific colleague here at the center. The intro, VIJAY, that you gave, I was very appreciative of that. You really phrased what we're after here. The technology is really interesting. The science is really interesting. But at the end of the day, we're doing this because we want some outcomes that matter somewhere. And those outcomes, for example, in Ivan's research or in the labs of others here at the center, those outcomes are improved crops that yield more with less water, less fertilizer, and less land. That's, VIJAY, I can't say it any better than you did at the outset. That's what this is all about. It's not an instantaneous process. That is to say, there's a lot of work that's going to go in over a long period of time. It's been going on for quite some time. And we're seeing outcomes now in the field that are the result of this type of research. It's a really exciting time in plant science. Now, Chris, if you can only tell me why Mike Adams can't lay a bunt down the third base line, I'll be thoroughly happy if you can answer that question, because it could have made a big difference in the recent season.
I expect you'll see that more next year.
So let me finish by thanking Molly and the Conversations group with the Friends Committee. Molly, you've hit a home run again with your selections. We look forward to the next conversations. We're thrilled to host these. We hope you all come back. Have a good evening and safe drive home.